Welcome everyone to our presentation of CureGRIN's research roadmap. Our agenda for today is Megan and I are going to start by taking you through the roadmap. We're lucky to have Dr. Adi Barzell here today. He's going to talk about gene therapies. He advised us on the working group when we were developing our plan for gene therapy. And then Denise is going to talk about how you can help, whether you're a researcher or a clinician or a family member. And then she's also going to give a CureGRIN update. And then we're going to finish off with a question and answer or discussion session. So if you have any questions, put them in the chat, or you can raise your hands at the end. We are recording the session, so if you ask a question at the end, you are consenting to be part of that recording that we're going to share afterwards. So we called our research roadmap Treat the Symptoms, Cure the Disease. And this is based on some of the feedback that we've received from families that we'll take you through as we go along. So our goal in developing this roadmap is to identify and prioritize specific questions that need to be answered in order to develop cures and treatments for GRI disorder. So I'll tell you a little bit about what we mean by GRI disorder shortly, but essentially it's GRIN disorder and other related GRI genes. And we want to do this for two reasons. We want to define how we as CureGRIN are going to invest the funds that you donate or corporations donate or other organizations donate to us, how we're going to donate research and science. But we're also hoping that this collaborative roadmap will define priorities for researchers, for biotech companies, and for other GRIN advocacy organizations. We kind of started this process when we got our grant. Actually, when we were preparing to get our grant, applying for our grant from CZI, we learned about David Fajgenbaum, who is known as the doctor who cured himself. So he had Castleman's disease, was dying, and basically started researching drugs that were already available, so FDA approved drugs, and found one that actually was able to put his disease in remission. And as part of the Castleman family or patient organization, he developed this new model for how he says that, and it's been embraced by CZI, model for how groups like ours should operate. So the traditional model is that groups would go out and fundraise, and then they'd do an RFP or an RFA and ask researchers to come forward with ideas or suggestions of how to spend the money. And then they would sort of look at them all and say, okay, this one sounds more worthwhile than this one. And what the shift that we're seeing now based on his recommendation across patient advocacy organizations is that we're moving to this new model where we really start with collaboration between families and researchers to define research priorities and these important questions. And then we fundraise, and then we go out and find the best people for the job to answer these questions. So some of them may be people who we already know, who are part of our scientific advisory board. Some of them may be people that we haven't been in contact yet, but they have a specialty that ties in with what we're doing. And in some cases, we may just not know, and so we will do RFPs or RFAs for some of the questions. So our roadmap journey started with a detailed research audit that Megan Collins prepared. And we're going to be publishing the final version of that this week. We consulted families with a survey and focus groups, and I'll share some of that data today. We consulted with researchers and clinicians through surveys and our research roundtable meetings. Then we created a working group of stakeholders, both researchers and families. And then we went through an elaborate process to identify the essential questions, 10 questions. Then we consulted with stakeholders, went back to researchers and to the different family groups to get any final feedback. And so we're here today with this presentation of the roadmap. And I'll get to the next two steps later. So here were the members of our roadmap. So along with me, there was Dr. Trinellis, Dr. Ramsey, Dr. Frankel, Dr. Barzell, Megan and Jillian from CureGRIN, Liz and Carol from the GRIN2B Foundation in the US, and Sandra and Amela from GRIN2B in Europe. And then we also had a GRIA family representative, Heather Cartwright, who's been sitting in on our GRIN board meetings. So when I talk about GRIA and GRI, when we first started CureGRIN, I think it was a bit of an evolution, because before that, I think many of us as patients kind of thought of our diseases on our own. So we thought of GRIN1 as something very separate from GRIN2B and GRIN2A. And so our vision was to bring it all together in one group, because we saw how similar all of these were. And so that was kind of the idea in founding CureGRIN. And as we've moved forward, we've learned more about all of the similarities between GRIN and NMDA receptors and these other receptors, and especially the AMPA receptor, which is coded by GRIA genes, and the kainite receptor, which is coded by GRIC genes. So we've been in contact with a number of families with GRIA disorder, and there's also a couple families plugged in with GRIC disorder. So as we connected with them, as we talked to researchers and learned more and more, really the feedback that we got from researchers and others in the space was that we would get to cures and therapies for GRIN disorder faster if we worked together. So if we thought of our science plan as not just something for GRIN disorder patients, but for all of these GRI patients, because there would be, there's so much overlap, so much potential similarity in terms of treatment options. Almost all of the genes are known to be connected to autism, so there's similar opportunities for research funding. And we've also had GRIA families that have been raising funds for CureGRIN. So we decided that we wanted to make all of the GRI genes in scope for this project. And CureGRIN, we're also going through a process right now where we are trying to decide as an organization if we formally, in addition to just having them, as part of this process, if we formally want to evolve and kind of represent all of these genes moving forward. So we did a family survey. We had 197 responses from GRIN families and 15 from GRIA families. So you can see on the left here, we had good representation from around the world. Over 60% of the families were from outside of the United States. So I think that really bodes well. It shows that GRIN really is, obviously an international disease, and CureGRIN is so far being successful in setting ourselves up as a global organization. And then this is always just, I think, interesting to share that in terms of the parents who filled in the survey, 90% of the patients that we're aware of are under 20 or 20 and under. So there's a very small patient population that is older than that. And we think the reason for that is just around diagnosis, that people when they're young children are more likely to have genetics done. This genetics testing is new. So we know that more and more young people are getting diagnosed, and it's not really catching up to those kind of especially older than 30, where the older you get, perhaps the less your parents are involved in your medical care, the less often you're seeking doctors, and the less often you're looking for diagnoses. So this is one of the most important pieces of feedback that we got from families. We asked them about how we should be balancing cures and treatments moving forwards. And so if you think of a cure, it's something that could resolve most of the symptoms of CureGRIN. It could, help someone get to, closer to a neurotypical child and have a lot of symptoms remedied. And that would likely involve gene therapy, although it's also possible that drugs that target the receptors could result in something like a cure. A treatment we think about as something that could impact one symptom or perhaps two related symptoms, and wouldn't be gene therapy likely. It would likely be some kind of small molecule, some kind of drug that could target the NMDA or the receptors, but could also target other systems. So if you think of that, something like treatments, drugs for epilepsy that many GRIN and GRI patients have. So I'm just going to go through these next slides really quickly, because this is kind of a summary for each of the genes in terms of the left column is what they said was the most important, the most common symptom. So you can see that for GRIN1s here, intellectual disability, speech, and sleep challenges, the ones in red, were the most common symptoms. And then when we asked, of all of these symptoms that your child has, if you had to pick one, what's the one that you would really want to see a cure or treatment for? And here we saw, some differences between the different genes, but for GRIN1s, it was intellectual disability, epilepsy, and neurostorms. For GRIN2A, the most common were intellectual disability, speech, and epilepsy, and the most common, and then the most important for that group was epilepsy, by far as the number one, followed by intellectual disability and mood and behavior. For GRIN2B, the three most common symptoms, intellectual disability, speech, and low muscle tone, and the most important, intellectual disability, speech, and mood and behavior. And for GRIN2D, where we had a very small sample, there's not as many known patients with GRIN2D. So there were, I guess, of the 10 patients that responded, or the families that responded, 80% of the children had intellectual disability, 80% had low muscle tone, 80% had epilepsy, and 80% had mobility impairment. And when we asked them about the most important symptoms, again, epilepsy was the dominant one, followed by intellectual disability and mobility impairment. And then for GRIA, so here again, it was a small sample, but about 15 people. And the top three responses here were most common symptoms, intellectual disability, speech, and mood and behavior. And the most important symptom to help was mood and behavior and epilepsy, followed by intellectual disability. And I think I see some of you taking notes. So just so you know, we will be publishing all of this online or on the website this coming week. So all of the information, actually a lot more than we're presented today, will be there. The full research plan will be posted. And then we also had focus groups. And we learned more about the symptoms and the priorities, but there were also a couple of other things that came through in these focus groups. So families told us that what they were prioritizing the most important symptoms, one of the key things that they were really focusing on was the quality of life. And we heard from a couple of parents who said that things like physical disability, like not being able to walk, or even a visual impairment, that they believe that people can have good lives with these kinds of impairments, but with severe seizures or neurostorms, it was much harder to have a good quality of life. So that was part of what influenced how they answered the questions. They also told us that with epilepsy treatments in particular, they knew that there were lots on the market, but so many were having trouble finding ones that worked for their children. So if there was a way of knowing which would work best for GRIN disorder, then that would be really helpful. And families also wanted us to really remember that all of these symptoms, or many of these symptoms, are interconnected. So kids with visual impairment makes it harder for them to learn to walk. And kids with severe gastro pain, that can get in the way of their ability to learn. And then when we talked about the symptoms, the feedback we also got was that there was some overlap between some of these symptoms. And so it allowed us to kind of consolidate our categories. So what we came up with from all of that were three tiers of priority for symptoms. And this is what we're focusing on in this plan. So the top three were intellectual disability and communication, which we combined into one category, epilepsy, and then two other categories that we brought together. So mood, behavior, and neurostorms. And then we have this second tier with feeding and digestion related symptoms, with mobility symptoms and sleep related symptoms. And then in the bottom, we also have things like breathing, low muscle tone, vision, hearing. So that's kind of how we decided to focus moving forward was based on that feedback. So the other thing, before we got to the 10 essential questions that we wanted to talk about was sort of how we would allocate our time and resources into 3 potential routes for treatment. So I've already mentioned gene therapy as one route. Another route is drugs that specifically target the NMDA receptors. And then a third category is drugs that target symptoms and could, that could be like an epilepsy drug. It could be a drug that targets other downstream symptoms. And so we decided in talking to researchers and other family members that our top area of focus is going to be gene therapy for about 40% of our effort, followed by drugs targeting NMDA receptors and drugs targeting symptoms, each at 30%. And so I think I'm going to turn it over now to Dr. Barzell, who's going to talk about gene therapy.
Hi, everyone. Can you hear me well? Great. So I'm very happy to be here with you today. Perhaps some background. I'm now a researcher at Tel Aviv University. Previously, I did my postdoc at Stanford University. And I'm also a co-founder of a couple of companies. One of them is called Logic Bio, and it's interested in rare genetic disorders. We are now in a clinical trial in methylmalonic acidemia. And in general, we are trying to treat diseases that can be treated by targeting the liver. So quite different than GRIN and GRIA, but hopefully I can share some of my background, some of my knowledge, and perhaps together we can think how these could be implemented also in the diseases that your children are affected with. So gene therapy is here. It's no longer some far-fetched dream. As you may know, some 10 years ago in Europe, the first gene therapy was approved. That was for LPL deficiency. And since then, we have had several treatments being approved for spinal muscular atrophy, for Leber congenital amaurosis, and also in several types of leukemias and lymphomas. And you may also know that the recent COVID vaccines are in fact some kind of gene therapy. So some of them are based on mRNA, some of them are based on viral vectors, which is an important concept, which is also relevant here, so I will explain it. A viral vector is something that used to be a virus, but we know how to engineer it, so it will serve us as a vehicle to bring into the desired tissue and into the desired cells the recipe with which the cell can make whatever is missing in that cell. And this is the concept used in several of the COVID vaccines. It's also the concept used in SMA gene therapy, in Leber congenital amaurosis gene therapy, and something that I would recommend trying also in GRIN and GRIA. Nobody can promise that it would have an effect. It's something that has to be tested thoroughly, first in animals and later in clinical trials. But I think it's worth a shot. So in particular, what is used in several of the treatments I've described is a particular viral vector. It's called AAV. AAV stands for adeno-associated viral vector, something that used to be a virus, but even as a virus, AAV was non-pathogenic. It didn't cause any disease, let alone after we engineer it. So it's a relatively safe vector. It's not 100%, and you shouldn't put too much of it. It can be dangerous, but a relatively safe vector. What we do is we take out the viral genes. We only keep the capsid, which is the outer shell that allows this vector to go into the desired tissue. And inside that capsid, we now put the genes of interest. In your case, it would be the GRIN genes or the GRIA genes. there is a capacity limit, which would mean that for some of the green and GRIA disorders, it would work with AAVs. But if it's too big, one might opt for another viral vector with a larger capacity. And each viral vector has its pros and cons, but the concept is similar. We put our desired genes inside a capsid that would take them to the tissue of choice. In your case, the brain. In some of the green and GRIA disorders, we might want to do more than just provide the correct gene. We might also want to knock down the harmful gene. We don't know, and I'm far from being the expert, there are much greater experts than me in this panel. In each and every case, whether the malfunctioning allele, which is the malfunctioning version, is also doing damage. But it's very possible that in some individuals, it's not only missing, but it's also doing damage. In this case, we would want both to provide a good version and also to knock down the bad version. And we do that by coding inside the same capsid also a small chunk of RNA that would cause the malfunctioning gene to be knocked down. In fact, it might and very well probably will knock down both versions in the cell, the normal version and the malfunctioning version. But it's not too bad that we are knocking down also the good version, because we are providing a good version from the outside, and we can provide it with desired quantities. It's not an exact science, but we have some ability to control how much of it would be expressed in the cell. So we can try first in animals and later in humans to get to the right level to be expressed in the cells. So what you're seeing here now on the slide is the concept of knockdown and replace. We are knocking down the expression of both the normal and the bad version, but we are providing a lot, at least enough, of the good version in order to provide the function. So this is the concept of knockdown and replace. Keith, do you want to look at the sizes or, okay. Yeah, so we have here the sizes of the different genes that are involved in green and GRIA disorders. So definitely for green 1 and GRIA disorders, these could fit in an AAV. The other disorders, green 2s, would probably need a different vector, but other vectors are being actively pushed to the clinic. In particular, one particular other vector called an adenovector has been used in the recent COVID vaccines at lower doses, but I think it creates hopes that it could also be used at higher doses for gene therapy, and in particular also be tested for the green 2 disorders. So we can go one slide back and discuss the advantages and what can be done with it. So it's a potential single treatment. Again, I want to be very careful here. Nobody can promise that it would have an effect, let alone reverse things that have already happened during development. It's possible, but nobody can promise that, and it has to be tested first in animals and then in clinical trials. So it's a potential of single treatment for each gene, regardless of variant type. So it could be one drug fits all for each particular gene. So for green 1, one drug fits all. For green 2A, one drug fits all, regardless of the exact mutation. It's regardless of the function, whether it's a loss of function, so it simply is missing, or something that is dominantly negative. Still, this should work because we are knocking it down. And potential, again, I'm cautious, of reversing a phenotype of a target cell. The questions here, would it be very expensive and who would pay for it? So yes, gene therapy is very expensive today. I hope that like any technology, with time, the prices will go down. We don't know which symptoms will be reversed and how age dependent this is. I think the delivery is the main issue, and my recommendation to the committee was to invest a lot in how to deliver the vectors to enough cells in the brain. I see this is the major challenge. How to deliver larger genes. So we discussed we may need a different vector for larger genes, and how long to get regulatory approval. This will take quite a few years. It's possible. It's faster today than it was 5 years ago, but it's still not trivial. But in order to get to the finish line, one has to start. And I definitely think that this is a promising route to embark on. I'm happy to take any question.
Thank you, Dr. Barzal. So if you do have any questions, you can put them in the chat now or put up your hand at the end of the session. And so we'll just keep going for now. One thing I'll just mention, though, is that we are also excited to welcome Dr. Barzal to our Science and Medical Advisory Committee. He's agreed to join us, so his expertise will be really valuable there. So thanks. We look forward to continuing to work with you, Dr. Barzal. So a couple more slides before. Oh no, actually not. Yeah, so I'll hand it over to Megan now, who will take us through the 10 questions.
Hi, everyone. It's such a pleasure to be with everybody today. So I am going to be walking us through our 10 essential questions. So as Keith mentioned, it's been a long process to get to this point. We've had lots of colleagues to advise us. We've been working with families through the working groups. So it's super exciting to be able to share with you a little bit about what we want to invest in and what we want to research. So our very first question is, what are the right outcome measures? So we really want to make sure that researchers are measuring things that are going to be the most important to us. By agreeing on a set of outcome measures, research from different sites can actually compare studies together. So there are different, we want to find out what are the different suitable outcome measures to determine benefit from treatment. So essentially measuring things pre and post treatment, and how things should be measured for future preclinical studies and clinical trials. So some examples of some outcome measures to think about would be things like reduction of seizure frequency. So you would measure seizure frequency before treatment and then after treatment. Other things like assessment scores. So there are tests like the Vineland test, which measures behavior of intellectual and developmental disabilities, autism, developmental delay, the Wechsler Adult Intelligence Scale, which is going to measure cognitive ability in older adolescents and adults, the Social Communication Questionnaire, which would measure things like autism spectrum disorder symptomology, the Gross Motor Function Measure, which would measure changes in gross motor function over time. And some of these outcome measures are currently being studied in the ongoing L-serine trial with the Barcelona group. And they're looking at some other measures that we could potentially be studying as well. So really we want to identify what are going to be the most important outcome measures as we move forward to consider treatments. I want to go to our next question, Keith. So our next question is, can we find biomarkers? So biomarkers are essentially biological, chemical, or physical parameters that can be measured in the body. So we want to know, are there ways that GRIN disorder can change blood or other biological functions that could be reversed with treatments and cures? So can we find biomarkers in blood, urine, skin cells, cerebral spinal fluid, brain waves, et cetera, to indicate if a treatment is working, or even to potentially diagnose patients a little bit earlier? So biomarkers can be measured as indicators of normal processes, pathogenic processes, or indicate responses to treatment. So some examples that we might want to consider for looking for biomarkers would be things like seizure likelihood, where you could place a device and to see if a seizure is likely to occur soon, things like oxidative stress, psychological stress, and metabolics and nutrition. on to our next question. So question 3 is a cure possible at any age? So we've already talked a little bit about this. We want to know, is treatment's going to only affect young children or teens or even adults? So there's a really exciting study that was published by Dr. Amy Ramsey's group with the University of Toronto, where they looked at an adult loss of function GRIN1 mouse. So basically, they were able to take a look at this mouse using gene editing and rescue the phenotype. So in the image from their figure, we can see, so the wild type mouse, which you can see in panel C, this is what the normal expression patterns would look like. And then you have the rescue mouse, where you see that some of that is actually rescued. So really what this study suggests is that it's possible to reverse the developmental deficits that are caused by the changes in NMDA receptor expression. And this can lead to changes in the brain wiring. Also, this study suggests that there is the possibility to reverse the developmental consequences that could happen in adulthood. So really, for this particular question, we want to see if we are able to reverse deficits and also better understand the importance of the timing of treatment. So when is the right time to start therapies? Prenatally, can there be positive effects regardless of when therapies start? How can we actually delay or possibly prevent symptoms? And are the symptoms reversible or essential to provide a treatment before they appear? Okay. Oh, I think we skipped one. Okay, so question 4 is, what's the best delivery route for gene therapy? In the spinal cord, to a specific region of the brain? What is the best method to administer gene therapies? So we've all learned that our nervous systems are very, very complex, and there are certain obstacles to delivering the ideal gene therapies. Some of those obstacles include the blood-brain barrier and how invasive a particular delivery method might be. So in this top picture here, we can see an example of what the blood-brain barrier looks like. So basically, our blood-brain barrier, it's a barrier between the blood vessels or the capillaries in the brain and cells and other components that make up brain tissue. So like our skull and our meninges protect against physical damage, our blood-brain barrier is going to be our internal defense system, protecting against pathogens and other toxins that might be present in blood. And so there's a tight gap that only allows for small molecules to fit through, and some gases. And then there's also some other molecules like glucose that are larger that can go through other ways. But basically, the blood-brain barrier is very effective at preventing unwanted substances from getting into the brain, which can have its challenges. So a lot of potential treatments don't readily cross the blood-brain barrier. So we have to investigate different ways to have these gene therapies delivered to the brain and in a large enough quantity to the right areas so that we can see if there are benefits from treatment. So some examples of different types of administration that we would like to study are things like intravenous, where we could inject medicine into the vein. So for example, in that bottom image there, where there was an AAV, they injected it into the vein, and then it goes through the bloodstream to the brain, and is able to deliver to the central nervous system there. There are also other forms like intracerebroventricular injection, which is like where you would inject it into the CSF, intraparenchymal, where you would inject it into the gray matter tissue in the brain. So some of these different delivery methods are a little bit more invasive than others. So that's definitely considered an obstacle, and we want to figure out what's going to be the easiest and safest way to deliver gene therapy. So our next question is, how can we deliver gene therapies for larger genes? So Dr. Barzell and Keith already talked a little bit about this. So the larger genes can actually be more difficult to deliver gene therapy. So we actually only know of 2 FDA-approved AAV gene therapies, Luxturna, which was approved in 2017, and Zolgensma, which was approved in 2019 for a rare, the first one was for a rare inherited retinal dystrophy, and the other was for a spinal muscular atrophy. And so we know that the GRIN1 and GRIA genes are going to fit into the AAV, and we want to understand what's the best vehicle to deliver the GRIN2 genes. So we've talked about how some of the genes are too big to fit into an AAV, so we might want to pick a different vector or use another type of technology. So we're going to work with biotech companies and conduct research to assess what's the right strategy for gene delivery for these larger genes. So our next question is, what are optimal drugs and molecules targeting NMDA receptors and related ion receptors? Can drugs bring GRIN-related receptors into balance? So I wanted to walk everybody through. I know we have a bunch of parents on the call who might not have heard of some of these different types of drugs, so I thought I would walk you through that. So the first one is an agonist. This is going to be a substance that's actually going to bind to the NMDA receptor or AMPA receptor and activate the receptor, and it's going to have a biological response. For NMDA receptors, a typical agonist could be thought of as glutamate. And then there are different types of agonists. So you have things like endogenous agonists, which are naturally produced in the body that are going to be things like hormones or neurotransmitters. Exogenous agonists are actually going to be external factors. So thinking about things like drugs. And then we have things called antagonists. These are going to be drugs that interfere or inhibit physiological action of the receptor. So a traditional NMDA receptor antagonist to think about is memantine. And there have actually been lots of studies, N of 1 studies and smaller group studies that have been looked at for memantine use. And numerous studies have shown in GRIN gain of function patients that there's a reduction of seizure burden from memantine treatment. So there was a paper published this year by Dr. Trinellis and his colleagues looking at a GRIN1 gain of function where memantine reduced seizure burden. GRIN 2A gain of function study was published in 2014, as well as I believe last year in 2020 that showed a reduction in seizure frequency. And then for GRIN 2B gain of function, it was used in 4 different patients, but I don't believe they saw many changes in terms of the seizure frequency in those particular patients. So definitely something to consider as a potential treatment. Other drugs such as PAMs and NAMs. So PAMs are positive allosteric modulators that are going to act to enhance the function of a receptor. So PAMs actually work in the presence of an agonist. So that first one that I was talking about, the PAMs are going to work in the presence of that. And then NAMs, these are negative allosteric modulators, which are going to block the binding of an agonist to a receptor. So they're going to bind to a site that's different than the spot where the agonist binds, and then you would see if there's a functional effect. So we're hoping to explore some of these options as potential treatments to potentially affect some of the symptomology and maybe bring these GRIN-related receptors back into balance. So our next question is, are there approved or late-stage drugs that could be repurposed for GRIN and related GRI disorders? Could there be drugs already out there that will help? So essentially here we're talking about doing something like a high throughput screening, which is basically where we use bioinformatics as shown in this image. So you would start with bioinformatics and target identification to think about different things that could potentially affect this particular receptor. Then we would go into in vitro studies, which are going to be using cell cultures and things like that, go into assay development, and then do all of these screening libraries where we would look at FDA approved drugs, other molecules, and taking a look at certain drugs that maybe didn't make it past FDA approval. So that might have stopped earlier in the process, and to screen those things and then do in vitro and in vivo assays. So looking at animal models as well as cell models, and try to see if there's going to be a good lead. So something that could go to clinical trial and potentially approach the regulatory process for FDA approval, and then would be go through the post-market surveillance afterwards. So our next question is, which symptoms are due to receptors outside of the brain? So we know that GRIN genes are highly expressed in the brain, but they're also expressed in the gut, the lungs, the nervous system, all over. So again, the highest expression is in the brain, but they are expressed outside of the brain as well. So this image is kind of small, hard to see, but basically all of these little lines down here are showing NMDA expression, so these little bar chart like things, and we see that the highest expression is going to be in the brain, but there's also high expression in the heart and other parts of the body. So there's some low level expression elsewhere, but really what we want to better understand is some of the symptoms caused that are changed in the NMDA receptor. Are they caused by something happening in the brain or outside of the brain? For example, with hypotonia, is this caused by NMDA receptors in the brain or in the somatic nervous system? Also, are respiratory issues caused by hypotonia originating in the brain neurons, or is it localized in the respiratory system? So there was actually a study published this year that looked at the expression of NMDA receptors in the human pulmonary artery. And the pulmonary artery works to carry deoxygenated blood, so blood without oxygen, from the right side of the heart over to the lungs. And so there has been research showing that NMDA receptor subunits are actually expressed throughout the lungs and the trachea. And this particular cell type, if there's too much activation of the NMDA receptors in the lungs, this can actually lead to something called acute nitric oxide dependent injury. And so CureGRIN has actually been notified of a few small number of unexpected events related to respiratory issues in our patient community. So this is one really important question outside of the brain that we want to consider and potentially invest in research for. Hope everybody's still with me. We're almost there. So question number 9, can we improve symptoms targeting downstream? So here we're wanting to ask which symptoms can be ameliorated by combinations of substances targeting downstream effects. So I'm going to go through each one of these and kind of explain what they are. So the first one is oxidative stress. So this is basically like an imbalance of free radicals. So these are oxygen containing molecules that have these electrons and also antioxidants in the body. So these can actually lead to cell and tissue damage. Another potential downstream effect that could be explored is neuroinflammation, which is essentially a basic immune response of the cells in the brain. So there's a concentration of inflammation that protects our neurons or our brain cells from damage. But if you have too much neuroinflammation, this can intensify neuronal damage. So that could also be something to study. Mitochondrial dysfunction. We've all heard of mitochondria from our high school biology classes being the powerhouses of our cells. But basically, if you have mitochondrial dysfunction, this is going to happen when the mitochondria isn't working as well as they should. And this can be due to things such as environmental factors or genetic abnormalities. So something to consider there. Nutrient signaling dysfunction. This can be things from like IGF, like growth factors, mTOR, which is the protein kinase mechanistic target of rapamycin. It's basically a cell growth regulator. So it's important for understanding cellular metabolism and cellular growth. And there's lots of environmental inputs that actually affect this particular complex. Some other things like excitatory inhibitory imbalance. So if there's too much excitation or inhibition, methylation and epigenetic factors. So epigenetic markers, they're basically things that tell our genes to cut on or to cut off. And there are 2 different types. You have chemical, which would be like methylation, and you also have protein, which are things like our histones. And so through these epigenetic markers, there are environmental factors like diet, stress, prenatal nutrition that can actually imprint on genes and be passed on. So we really want to explore if any of these symptoms can be ameliorated through combinations of treatment for these different things. And finally, we are here at the end. So our very last question is, what are the functional and phenotypic details for each variant? Functional analysis and natural history by gene and variant. So what we really want to understand here, a lot of you have already been through this process where you've had functional testing on your child's variant. So we really want to understand what are the functional and phenotypic consequences of each variant. So this would be continuing to do functional testing on every identified variant through groups like CFIRB, the Barcelona GRIN team, and lots of other teams around the world. Also trying to find improved diagnosis and effective treatments. So we need to better understand the functioning of GRIN and GRIA variants. This would be studying our animal models that we have from lots of groups around the world and all of the researchers who have been participating in our roundtables. Lots of them are very interested in these things, and we want to continue to work with them and partner with them to learn more about what's happening as a result of these variants. And we also don't necessarily know how the brain's architecture is changed by GRIN and GRIA variants. So we want to do further analysis on the existing animal models, continuing to do the registries and performing natural history studies, as well as setting up a brain donation program for GRIN angels to better understand different changes to the human brain. And so I hope I didn't lose anybody. I hope we made it through successfully, and we're very excited about our essential questions.
Thank you, Megan. So that kind of wraps up our science plan. And I'm going to tell you about our next steps in a minute. But first, Liz Marfia-Ash, who's the head of the GRIN2B Foundation in the US, just asked us to share this with the group. So their organization's been closely involved with this project. And so you can see there that she says that on behalf of GRIN2B, they endorse it and believe that this research plan sets the GRIN community on the right path towards treatments and cures in the years to come. And so maybe I'll just pause for a minute. There are other people who were part of the roadmap committee that are also here. We'll pause and see if any of them want to add anything.
Yeah, hey Keith, I just wanted to jump in. This is Steve Trinellis, and I had been interacting with CureGRIN, and I just wanted to mention, first of all, Megan, terrific job going through that presentation and making it very clear for everybody. I wanted to say that I was impressed by the vigor and the high energy that the CureGRIN team brought to this, and I also enjoyed this comprehensive manner in which they really sought out advice from all stakeholders. And so I think this is just a terrific project. This is a great starting point, and I'm enthusiastic to see where this leads.
Thank you.
Yeah, I don't have anything to add except that I am continually impressed by how fluent the parents have become in this very technical language. And when I hear this, I'm always impressed by it. So I think that this community of parents and researchers is going to accomplish amazing things. And this roadmap is very helpful for researchers like me to see what's important.
Okay, thank you. Thank you so much. So in terms of what's next, so after the presentation, we are going to, this coming week, we're going to publish the full report. It's about 100 pages long, but we're going to be publishing that as well as the research audit that's, I don't know, another 40 pages long, I think. So those will both be on the website. We are also planning to publish blog posts with detail on each of the questions that'll come out over the next month or so. And we'll also be translating summaries, shorter versions of the report into other languages, and I think translating this session into other languages. And so we'll make it all available on our site. So then after that, the next two things are first, the 8th step in this process is to collaborate with stakeholders again and to identify the best path forward for each of the questions. So it may be different for each question. So for some, we may set up another question-specific working group where we'll bring back key researchers and family members together to talk about the best way to go forward. For others, there may be opportunity to partner with other rare disease organizations or pharma or biotech. So for example, when you think about the question of how to deliver effectively to the brain, where's the best place to deliver it, and how to deliver big genes, those are things that a lot of other patient advocacy organizations for neurodevelopmental diseases are also looking at. So there's opportunity to collaborate with them. We are looking for grant opportunities and working with researchers to pursue these. There's already 2 that we've identified that could potentially answer some of these questions. And so we're getting in contact with researchers to see if they want to be part of this. And then we expect to be able to award, so our plan is with the funds that we've already raised, mostly from families and from their friends. We've already got funds that we have at the end of last year, and then adding that to the funds that we are expecting to raise this year, we expect to have $800,000 available to give out by the end of our fiscal year, so next April 30th, and more funds in coming years. So we could award that much. We could award more if it includes funding for future years, but that's the plan. So which questions we have funding for will depend on some of these other steps. And then step 9 is to follow these paths and to answer the questions. And so our goal is for this to be a 3-year plan. So to have the answers by April 30th, 2024, which is actually the end of our 5th year for CureGRIN. So we've been at this for 2 years now. And so whether we can make the date or even exceed it are going to depend on a few factors. This is not something that Denise and Megan and I can do on our own. So we know a big part of this is going to be, the willingness of the GRIN community to help us raise funds. The success in CureGRIN and researchers in securing grants, and a commitment to collaboration across patient advocacy organizations, researchers, clinicians, and industry. And so far the collaboration has been really great, and we want to really continue this as we move forward to this new phase in our growth. So I'm going to turn it over to Denise in a second, but first I just want to talk to scientists and physicians who are on the call specifically. So how you can help us achieve this plan. Number one is to be essential. So what we mean by that is consider how the research that you're doing now could be nudged towards these 10 essential questions that we've identified. Number two is to keep us informed. So let us know how your research is going. Number three, work with us. So if you see a grant opportunity and you are looking to apply for something GRI related, let us know, because we think that we can help you. We know that at least one grant recipient has said that without family support, they don't know if they would have got it. So and we know that it's becoming more and more important for grant givers that families are involved. So please collaborate with us and collaborate with other researchers through our GRI research roundtables, which happen every 6 weeks. And then finally, just stay connected. So CureGRIN.org/newsletter. We have both a regular newsletter about our operations, and then a specific scientific newsletter that we send out. And we're also on all of the regular social platforms. So I'll hand it over now to Denise.
Thank you, Keith, Megan, everybody. This is truly amazing. I hope everybody is excited as I am about our essentially our roadmap for a cure. It's just amazing to see the progress that's happened over a very short time. So again, thank you to everybody. Thank you to the researchers. This is so exciting. So I wanted to share a little bit about how families can help us achieve our plans. So we've mentioned this before, we have different registries for different regions. Hopefully you all know about them, but if not, we provide the link there in the presentation that we'll be sharing summaries with afterwards as well. And also, if you have participated and maybe need to update some of the information, I think that's. And please mute your phone. I don't know if you can, oh, there we go, sorry. Okay, and then obviously this research is going to take a lot of money to produce, and families play a crucial role in helping us to raise the funds for these treatments and cures. So I'm going to be sharing a little bit in a few more slides, different campaigns that we have going on that you can get involved in. Also want to make you aware that our GRIN conference is coming up next year in April in Boston. We will be sharing more information about that in the next few months and how you can register, but make sure you save the date on your calendar for that. And volunteer if you have any special skills or talents that could help us along our path. Please reach out to us at our info email there, and we'll be sharing a little bit more about some other volunteer opportunities in the future. And like Keith said, stay connected with us. If you're not following us on Facebook, Instagram, Twitter, LinkedIn, be sure and sign up for the newsletter to stay up to date about all this great information. Next slide. And then we wanted to share a few highlights of our past year. Obviously, we've created the science and research plan that we've been talking about here, which is a huge undertaking. We've also been able to award CFIRB $75,000 grant to help with the functional analysis of additional families. And through a grant through Global Genes, we awarded 15 families $1,000 each that were impacted by the COVID pandemic. Last year, we were able to raise $236,000 in donations. That includes the Global Gene grants. And we were happy to announce that we secured the second year of the CZI funding, which was a 2-year grant for $450,000. So we were able to secure that second year funding. Keith mentioned the Global Genes, the GRIN Genes Roundtable that have been going on for the past, I think 6-7 months now. We've been able to partner with GRIA families and researchers to collaborate together. We produced an amazing Stories of GRIN book that was that many of us have enjoyed reading and sharing with our families. We've been able to produce a couple of conferences. Obviously had to go virtual last year, the virtual conference in September, as well as a mini Q&A that was just a few months ago, and have been able to translate those recordings into different languages, and we'll do the same for this recording as well. It's important for us to be able to reach our communities around the world. We have the amazing GRIN 2D video that hopefully most of you have seen. It's on our homepage of our website. It's an award-winning video, so we're very pleased to have that video. Let's see. Last year we did our first virtual event, or a challenge event, Get Moving for GRIN, that raised over $15,000, and I'll be sharing how we're going to again do that this year, actually coming up next month. We'll be doing another Get Moving for GRIN. Okay, so when we first launched CureGRIN 2 years ago, we set out to raise we set a goal of raising $3 million in 5 years. So we are at the start of our second year. And I've included our actual income over the last 2 years. Our first year with the grant from CZI that we had to recognize in the first year, we raised over $600,000, and that's amazing. Last year, we raised $238,000, most of it from individual supporters, individual donors. And that's truly amazing, given that we all know we were in a global pandemic and a very, very challenging year. So this year, from individual donations, we are looking to raise a revenue of $323,000, including some event revenue. And you can see we have goals to raise additional funding from grants and from corporations as well, and then also to continue our book sales. So $500,000 in the year. We think it's doable. We think we're really excited about this. You could see how the next couple of years will take us to our goal of $3 million in 5 years. So again, families are really crucial in helping us to achieve this goal. And there's many ways that you can fundraise, and we can help you brainstorm some of those ideas. There's several different campaigns that we have coming up that I want to share with you. I mentioned this is our second Get Moving for GRIN. It's a month-long challenge, and participants can pick their distance that they want to accomplish, a half marathon, full marathon, or ultra marathon in a month. We're not going to be, we don't require you to track and submit your miles. It's an honor system. And we'll be providing links to all these different opportunities after this presentation and in the future blog post. We normally do our Count Me GRIN campaign in November, and we're going to be doing that again this year. And that follows it. The Giving Tuesday is right at the end of November as well. And for some families that had already been participating in different fundraisers, it might be a challenge to continue to fundraise on that very important day. So we'll be sharing information about how you can get maybe a family member or a friend to choose CureGRIN as a charity to focus on for Giving Tuesday for us. We call them the Friends of GRIN. Every March, this will be our third year of GRIN Disorder Awareness Month. That falls right after Rare Disease Day on February 28th. We'll be doing that again this year. And this year we're really excited. We're hoping to do some in-person events incorporating some maybe some local events in your area on a specific day. If you're interested in maybe hosting something in your area, please reach out to me. We're looking for volunteers to help organize those local events to continue to build awareness for GRIN disorder. And then also I wanted to share about some information about some year-round opportunities that you could participate in. We know sometimes some people may not be available, or November may not be the ideal month for them to participate in a campaign. So we're opening up the Count Me GRIN campaign to really year-round. You pick what time frame to launch your campaign, you set your goal. We're going to continue to look for champions to raise $2,500 each. But you know, we don't hold you to that goal. It's really up to you, and every little bit helps to fund this crucial research. And then there's always Facebook campaigns all year round. They're really the easiest way to fundraise. And many people donate their birthdays or just any time of the year, a quick Facebook fundraiser. And then I hope everybody has chosen CureGRIN as their charity of choice on Amazon Smile. It's really the easiest way to help raise additional funds. We all shop Amazon all the time, so it's a really quick and easy way to help bring in additional funds. And then, Keith, I think, yeah, you can go to the next slide. So I mentioned Get Moving for GRIN, the previous slide. Sorry. I could talk about this slide here. So we normally do a grand prize for the Count Me GRIN campaign for any family that reaches their $2,500 goal. They get one entry into this grand prize. And then for each additional entry, each additional $500 beyond the $2,500, they get an extra entry. So we want to announce the grand prize for Count Me GRIN, no matter what time of the year that you do it, is a luxury suite at our upcoming conference in Boston. So we wanted also to take this opportunity for the families that are on the phone. For anybody that wants to commit today, we are going to give you 5 extra entries beyond your $2,500 goal, beyond any other entries that you might achieve through your fundraising efforts. So you get additional 5 extra entries to have a luxury suite and stay at the conference in style. Keith, if you want to go back to the previous slide, I just had Get Moving for GRIN, that one. Okay, so this campaign is up and launched. You can go in and register as an individual participant or a team. It's a $30 entrance fee. You can fundraise. You don't have to fundraise, or you could just get out and just do your distance, but we would love for you to fundraise if you're able to. There are this year t-shirts and yard signs that you can order that let your neighbors and everybody know what you're doing, and hopefully get awareness and support through that effort. So if you're wanting to participate, I recommend go ahead and ordering your t-shirt and yard sign now, because it does take a couple weeks to get in. Okay, thanks, Keith. So we also have revamped and updated our fundraising toolkit. We've had some amazing families do some amazing fundraisers, and we wanted to share some of those just to give people an idea of other things that you can do. We'll be sharing the link to that. That's on our website as well. It does include some sample content like fundraiser letters and some memory joggers to help you determine what who you can reach out to for your different fundraisers. And the next slide. Okay, so we've been talking about this for a little while. Hopefully you remember we've discussed through the CZI funding. One of the opportunities that we wanted to create was a collaboration forum where we would have 3 different areas for one for GRIN families to communicate and collaborate with each other, another area for researchers and clinicians to collaborate with each other, and then also a 3rd area where families can talk and collaborate with researchers and clinicians together. So we're working on that right now, and that is targeted to be live here in the next 2 to 3 months. So when that is ready, we'll be really excited to share that with families and with researchers, and we hope you all participate and join the community. And I think I have one more, so that's the end of my online presentation, but I wanted to introduce you to 2 people that have stepped up to lead the sibling support group. This is something that our community has really expressed an interest in, and we're so happy that Soni Mullen, she's a sister of Tristan, and Abby Pelletieri, she's a twin sister to Owen. They both are co-leading this group, and we want to introduce them and let them share a little bit about some exciting things that they have planned.
Thank you so much. Hi, everyone. I'm Abigail Pelletieri. My twin brother is Owen, and he has GRIN 1 disorder. I've been working with Sunny, Tristan's sister, to create a sibling group for everyone with a sibling with GRIN disorder. We want to have many different educational and fun activities for the siblings. And we would love for everyone to join our Discord, which is practically a large group chat where we can talk and have video calls. When joining, the siblings will have to make a quick free account, and the link will send them directly to our server. There is a minimum age requirement of 13 years old. So if the sibling is less than 13, then their parent or guardian could make an account for them. I will also put the link to the Discord in the chat, and we will be putting it on our Facebook group, which is called GRIN Disorder Sibling Support Group, or you could always email us at grinsibs@gmail.com with any questions. We're hoping to create a community of siblings who understand and can help each other navigate life with a sibling with GRIN disorder. Not only do we want to provide discussion and resources, but we want to create friendships and provide support to all the siblings. Sunny, is there anything else you'd like to add?
No, that was great. I really appreciate all the work that Abby's been putting into this. And we're just really looking forward to hopefully making contact with all of the siblings of all of your children. I can speak certainly as probably one of the elder siblings, fortunately, unfortunately, that we understand that there's going to be quite a lot of age differences here. So our first goal really is to be able to figure out who are the siblings. And so that's what we're going to try to figure out. And like Abby said, there is the email now, grinsibs@gmail.com. And our first step is just to figure out who all the siblings are so that we can then plan from there, figure out what the age groups are, maybe where the locations are, and really strategize after that. But I'm really excited to have this group because I think for especially the younger siblings coming up, it'll be really important for them to have this going forward. So thank you guys for the support. And, as parents, we're here to support you as well with your children. And there'll be some conversations in that as well. So we're looking forward to working with you. And that's about it. So thank you guys so much.
Thanks, Soni and Abby. We're really excited to see what you have in store for the group. And Keith, I don't know if you saw, there was one research-related question in the chat. So is this a good time to address that?
Yeah, so maybe I'll just stop the sharing of the presentation. But first, yeah, let me just echo Denise. I hadn't heard all the details about the sibling support, but I'm so excited about what Sunny and Abby are planning. So thank you so much for sharing that. That is fantastic. So let me just look. Did you see what the question was?
Yeah, there's one question on imaging agents that cross the blood-brain barrier. Is it possible to use the same mechanism for future treatments for GRIN?
Is there one of the researchers who's on the call that would be able to answer that?
Well, I don't, I mean, there are lots of different strategies to get things across the blood-brain barrier, and it depends on what you're trying to deliver to the brain. So you can design drugs so that if they have charge on them, like a positive or negative charge, like if you can think about a magnet having, you know, a positive or negative end, they don't go into the brain so well. But if you take away the charge, or you make them more like an oil than like a water, they'll go into the brain easier. So if it's a drug, there's ways that you can modify it. And they're even developing viral capsids that are able to go into the brain. So it depends on what your cargo is, but there are ways to get around this, and there's a lot of science about that. Yeah.
So Allison wants to ask a question.
Thank you. Thank you, everybody, for all the wonderful presentations. My son has GRIN 1 and he has an autonomic breathing disorder. We have tried numerous drugs over the last few years. Our respiratory consultant has now devised, as part of the difficulty has been measuring what improvement in his respiration the drugs make, because it's so erratic. He's now devised a specific test for him to undergo, to then that we can then measure whether or not the different drugs that we've been trying actually improve or don't improve, or what, you know, so then we can make clinical decisions as to which way to go. He goes cyanotic and stops breathing and then kind of self-regulates and comes back. Is there anything that we can do to link in to make that more scientific, or more I don't believe other parent, you know, I don't know, is there anything that we can do to link in to so that it's as strong as it can be, or that we make sure that we're doing it in the right way, that it can benefit more people? Because as far as I'm aware, it's super rare even within GRIN. So I don't know, sorry, that's it.
Allison, no, you can certainly get him connected with us. He can email me, Megan@CureGRIN.org, or Keith, and we'd be happy to have him join in on our research roundtables and discuss some of those things with other clinicians, and potentially be able to create a partnership that way, and, you know, move forward with measuring those outcomes.
Okay, great.
Okay, that's her hand up.
Yep. Hi. First of all, I just want to thank you all so much. I'm almost at the point of tears. I just can't believe what wonderful work you're doing on this, and after only 2 years. But at the risk of stepping on a political landmine and doing research that helped me find your organization, there's another one called GRIN Disorder Research Association or something. Is there a relationship between your two organizations? Are you familiar with this other one?
Yep, actually, I think Tally from that organization is on the call. Tally, do you wanna, if you're still there, do you want to introduce yourself?
Hi, yes, I'm here. Just it's time that my children are still awake, so I cannot put myself up front. We definitely collaborate. We have different, obviously we have different researchers on board and clinicians, and I myself is a physician, and the researcher, and I just appointed I have a new job now as a head of research in one of the centers of rare disease in my country. So,
congratulations.
Yeah, so I am willing to do collaborations with also biotech companies, and to support research as much as I can.
Yeah, so we have, I think there's often kind of horror stories from other rare disease organizations that don't get along in a space, and I think we're pretty lucky. There's lots of great people working in this space, and I know Tali was generous enough to give us to answer. Give us some feedback on the key questions that we should be looking at and that kind of thing. So yeah, we really try to work as closely as possible with everybody.
Okay, great. That's wonderful.
Also, I'm hoping that now in the hospital, I have more power and more ability to support clinical trials and make, when we find the treatments, to make it into the clinic. Because there's also a lot of regulation issues that needs to be handled before you can start treating your patients. Thank you.
Thank you, Tali. Actually, I just want to, before we see if there's any questions for anyone else, Denise, well, Lauren just shared a link actually in the chat to the CureGRIN 365. Is going in there and setting up a page, is that the way that people will qualify for those extra 5 entries, or is it enough just to put in the chat that they're signing up, or email us today?
Either one. If you want to just put in the chat, raise your hand. We will try and capture your response. Allison's Val, okay. I see you, Val.
Awesome.
Yeah, we're still in the process of just making sure everything's correct on the campaigns, but yes, you can absolutely go and set it up.
Any other questions or thoughts?
I do have one, but I don't want to hog the whole thing. I'm wondering about the role of movement therapy in treatment or cure. I didn't really hear anything around that, and I know my kids are doing an awful lot of physical therapy with my GRIN1 granddaughter. She's hypotonic, so she's
yeah. So
is that effective? Is it helpful? Is it part of the process?
Yeah, I think there's a number of different kinds, and many of them can be very helpful for kids. I know a lot of kids have benefited from physiotherapy and osteotherapy and some of the other treatments that are available. I think we didn't include it in the plan because those treatments are generally already available and working. So we were focusing on areas where we need to commit to new research. So if there was opportunity to do research around that, then potentially it could be in scope as well. But the fact that it's not on the list doesn't mean that it's not a good treatment option. And so, the same with speech therapy, with vision therapy. Okay, well, thank you all so much for joining us on a Saturday for this. We're really excited about this next chapter, and we'll be sharing all the details on this next week. And as I mentioned, lots of blog posts coming up. So I see some people in the chat here have already committed to CureGRIN. So anyone else who wants to put their names in there before we sign off, go ahead and do that. But otherwise, I'll say goodbye. Thank you.